So first, let me thank you for coming out and fighting both the parking and the rain. I know it's a little difficult to get here. But today, what I'd like to do is talk about how animal research can help us understand the human brain. And much like in The Wizard of Oz, where they pulled back the curtain and you saw how the great wizard actually worked, I want to pull back the curtain on science, because I think for most people that don't do science on a daily basis, they don't exactly understand how animal research is done and how we can do experiments with mice or other types of organisms and learn anything about the brain at all. In fact, a couple of years ago, Sarah Palin was famous for saying that all of those investigators that used fruit flies to understand and advance human health, that was a complete waste of money. I guess she didn't know that several Nobel Prizes have been given to investigators using fruit flies as a model system. So I'm a professor in pharmacology, molecular physiology, and biophysics and psychiatry, but I wear some additional hats at Vanderbilt. I'm associate director of the Brain Institute, and what will become important at the end of my talk is I'm chair of the Institutional Animal Care and Use Committee and the director of the Office of Animal Welfare Assurance. And it's these two entities that are responsible for all of the animal research that's being performed at Vanderbilt to make sure that it's performed with the welfare of the animals in mind and that we are compliant with all of the federal regulations relating to animal research. Okay, so in my own lab, I'm a molecular neurobiologist. I'm interested in how the brain works, how cells in the brain communicate with one another, and I'll tell you a little bit sort of in the middle of the talk about some of the research that's going on in my lab. But obviously, everybody is interested in neuroscience research. Here, from the last 15 years or so, I pulled the covers of Time magazine, and these are all related to various aspects of the brain. So clearly, it's in the psyche of Americans, if not people throughout the world. In fact, when I was looking at all of these covers, I couldn't fit all of them in a single slide. So here's another one. I did want to point out, I wish I had read Secrets of the Teen Brain. My daughter just is about to turn 11, and I don't think scientists, no matter how smart they are, will ever figure out the teen brain. So studying the brain, in addition to being in the forefront of people's minds, no pun intended, it's interesting because if you look at the top 10 disorders that are leading causes of disability, and this was tabulated by the World Health Organization back in 2005, 7 of the top 10 causes of disability throughout the world are related to dysfunction of the brain. I won't go through all of these, but obviously the major cause of disability is unipolar major depression. And each year, there are 6.7 million people years that are lost as a consequence of major depression. So obviously, we have a lot of work to do to figure out the molecular basis, the physiological basis underlying these disorders, and then try and develop treatments or cures, I guess we'll go with treatments, to address them and improve the quality of people's lives. Okay, so the first question is, why do we need to use animals at all for research and teaching? So it turns out, and probably most of you aren't surprised, that the functions of cells and organs are basically the same in animals and humans. If you look at the biology of the mouse, and we'll go into this in a little bit more detail, the way that the liver works in the mouse is the way that your liver works, the gastrointestinal system, vision, all of the major physiological systems, down to the molecular level, they're basically identical. So they do serve as good models. And what we learn for these animals is useful for both treating human disease, but also for treating disease in animals. So when you go to the veterinarian to have your dog vaccinated, or treated with medication or a procedure performed. All of those procedures were developed using other animals that came before them. So I think I just want to convey it's not just human health, but it's animal health as well. Okay, so we need these animals because we're trying to understand how diseases affect living tissue. We use animals to develop and test treatments. So every drug that you've ever taken, well, let me rephrase that. Every prescription drug that you've ever taken was tested in animals long before it ever made its way to people. And any procedure that's ever performed in the hospital or at the doctor's office was worked out in animals first. The first hip replacements were performed in dogs, liver transplants, coronary bypass surgeries. All of that work was colonoscopies. All of that work was developed in animals long before it ever made it into the clinic. And you also need this to train future scientists and healthcare professionals. There are a lot of animal rights activists that feel that there is no difference between the quality of or the value of a person's life and the value of an animal's life. And I can't argue with that. That's just a personal opinion. I don't happen to share it. If we don't have animals to do these experiments, then our only alternative is to try these on other people. In the dark history of research, that has happened in the past. It has certainly happened in World War II under Hitler. There were a lot of people in the concentration camps in which they were doing medical experiments. At some time in the past, they were also doing experiments on prisoners. And fortunately, all of that is gone, but our only alternative is to use animals. If you have questions, rather than save them for the end, I'd rather have you raise your hand as we go, and I'll deal with them at the time. You know, it's called the Socratic method. You know, going back and forth, and Socrates taught Plato, and Plato taught Aristotle. So I think that was probably a good strategy for interactions. Okay, so the first question is, how do scientists choose an animal model to understand the brain? So there's a variety of animals out there, and which animal do you use? And it really comes down to what is the question that you're trying to answer. If you want to answer a question that just involves molecules, where most of the animals are all the same, it probably doesn't matter, and you would want to use an animal that's the lowest on the evolutionary tree. But if you want to use something, if you want to do an experiment to understand how perception works, or how reason works, or how making choices work, you can't really do that in primitive organisms. You have to do that in more complex species. And I'll go through some examples of these on the next few slides. Okay, so the first thing is that there are genetic similarities between all of the species through the process of evolution. From simple organisms, we developed and evolved ultimately into humans, but along the way, a wide plethora of additional species developed. And so this figure is just to show you the genetic sequence similarity between humans and other species. So it's interesting to know that what makes you 100% human, if I get the DNA sequence of one of you and then the person next to you, your DNAs are different by between 2 and 4%. That's what makes you, you, and the person next to you, them. Right? If we all had the exact same DNA, we would all be identical twins. Okay, so there's a 2 to 4% difference anyway. Okay, so some people use as a model system yeast. This is Baker's yeast. It's called Saccharomyces cerevisiae. It's used in the kitchen, but it only shares about 18% sequence similarity to humans. So for some questions, yeast are a perfectly good model system, but yeast don't have a nervous system, they don't have a brain, they don't have neurons, the cells that comprise the brain. So if you're really interested in neuroscience and how the brain works, yeast probably aren't the best example. Grapes, most plants share about a quarter of their genome with people. And there are a lot of things that can be learned from plants at the molecular level, but plants don't have a brain or a nervous system as well. Then you get into the animals, and here are 3 species clustered together. These are all invertebrates. These are animals that don't have a backbone, don't have a spinal cord. And we'll come back to a couple of these in a minute. The roundworm, the honeybee, and the fruit fly. And then we jump over to the vertebrates. So all of these animals have a backbone. We start with the chicken, but that's pretty much representative of most birds. The platypus, which technically is considered a mammal, but it lays eggs, so it's about as distant from the mammals as a mammal can be. And then we have zebrafish, mammals that we're more familiar with, and then mouse, and ultimately the closest to humans. Humans are primates, so these are other non-human primates. And at the top of that, probably the closest or near the closest to humans is the chimpanzee. Now, for those of you that are, please. The rat? It's about the same as the mouse. For those of you that are worried, Congress passed a law in 1986 that forbids the use of great apes, including the chimpanzee, for biomedical research. And the reasoning, and unfortunately, there are some things that the chimpanzee would be really great for because they're so close to people. But the problem is that they are so close to people, just because they can't speak our language doesn't mean that they don't have the same feelings and fears as everybody else. And so as you start using organisms that get closer and closer to people, I think that there's a concern, there's a worry even among scientists that that's not something that you really want to do if you can use one of these lower species instead. Okay, so the first example I'm going to show you is the roundworm. The Latin name is Caenorhabditis elegans, or C. elegans. Here's the worm. It's about 1 millimeter in size, so you can actually see it with your eyes, but it's pretty small, so here's under a microscope. This is what the worms do. They undulate, they swim around, they look for food, they eat bacteria. This is on the surface of an agar plate, but normally they're found in the dirt. So they live for about 2 weeks. They're 38% identical to people. Their genome, the number of base pairs of DNA in their genome is about 100 million base pairs, and they have 6 chromosomes. The reason that these animals are useful as a model system is they're pretty simple. In this entire worm, there are only 959 cells. And of those 959, a third of them are neurons in the nervous system. And these neurons, these 302 neurons, make connections with other neurons. And all of those have been identified, and there's only 7,000 connections. So if you wanted to make a circuit diagram for how the brain, how the nervous system of the worm works, you could do that because there's only 7,000 connections. This is in contrast to the human brain, where there are 86 billion neurons that make more than 100 trillion connections. So you're never going to be able to generate a circuit diagram with 100 trillion connections, but the 7,000 from this animal, you can probably do that. And at the molecular level, the molecules that are used in the nervous system are pretty much, if not identical, highly similar to what you find in people. Okay, so the next is another invertebrate, is the fruit fly, the Latin name Drosophila melanogaster. It's about 50% identical to humans, 3 millimeters in size. Now we go from 302 neurons. They have a much more complex nervous system. They have 100,000 neurons. Okay? And once you get up to this point, you can't identify the number of connections because it's estimated that each neuron makes 1,000 connections to other cells. Right? So that would be 100 million connections, and that's just impossible to map. Okay, the next is zebrafish. The Latin is Danio ririo. Now the size of the genome, 1.5 billion bases. It's about half the size of the human genome. It's 3/4 identical to humans. It lives for 2 or 3 years in captivity. These fish are fairly small, about 4 centimeters, so a little under 2 inches. And now these fish have 10 million neurons. They really have a brain. You would argue that although the flies and the worms have a nervous system, they don't have what you would call a true brain. But by the time you get to the fish, they have a brain and a nervous system that is quite similar to what you find in people. The major advantage to zebrafish is the fact that when they're embryos, they're transparent. So you can see what's going on during zebrafish development. So if you're interested in the development of the brain and where that can go wrong during embryogenesis, then this is a really good model to use. Yes? It's a good question. I don't know the answer to that. Supposedly, the dolphins, and this is just from the lay literature, I haven't actually read scientific papers on it, are supposed to be as smart, and I've even seen smarter than people. I guess it depends who the people are. Okay, so next we come to the mouse. Now, rats and mice represent well over 90% of all the animals that are used in biomedical research. So I'm showing you all of these other possible model systems, but more than 90% are mice or rats. They have a genome size that's almost equivalent to people. People are about 3.3 billion bases. They have 88% similarity. One of the nice things about mice is their gestation is pretty short. The dams are pregnant for only 3 weeks, so it's easy to generate a lot of mice pretty quickly to do biomedical research. The fact that they're small and you can house them in a fairly small space reduces the cost as well, so you have to be practical when you're doing the research as well. And mice have about 71 million neurons. Okay, so you're getting a pretty complex nervous system. Obviously mice don't have all the complex behaviors that people have. They do have a number of behaviors. They have foraging behavior and maternal behavior and mating behavior, but I don't think they worry about who they were going to vote for in yesterday's election. It just doesn't rise to some level of importance for them. Okay, so then finally you get to, please.
I toured a lab many years ago, and they were doing most of their work on cats. And it was on the brain, and they were one of those open house things they do every 150 years. So I was asking these questions like, why is it, what's the fascination with working on cats as opposed to mice?
There are some aspects of the cat visual system that are particularly amenable to experimentation. So there was some experiments probably back in the 1940s now where they would take newborn cats and sew their eyelids shut. Okay, just for a short period of time, and then afterwards ask, could they see normally or not? Did that affect the development of their vision? And it did. And so it provided insights for a number of human diseases where, related to blindness. And so that's why that's done. But cats are a pretty small percentage in the same way that dogs are a pretty small percentage. People, when people are sensitive about animal research, it's usually with fluffy animals that they would like to have as a pet. No one, I don't think anyone mourns for a fruit fly or a worm. That may not be true. But it's important that all of the animals, regardless, be treated appropriately and with respect. Yes?
When you receive a question from the audience, if you would repeat the question.
I will do that. Thank you. Thank you for reminding me. Okay, so at the top, the animals that are most similar to people are non-human primates. You could call them monkeys. Formally, they're non-human primates. Their genome size is about the same as people. They're greater than 90% identical at the genetic level. In captivity, they live much longer than out in the wild, about 25 years. This rhesus macaque gets to be about 17 pounds, and they have 10 billion neurons. Remember, humans have about 86 billion neurons. Now, these animals have probably as complex behaviors as people. They live in social groups, they have relatives, they have interactions, they have friends, and so they're a really great model, but they're rare, they're expensive, and if you can answer the scientific question using an animal that's lower on the evolutionary tree, then that is definitely preferred. It's only those very specific questions that you can only use with non-human primates that are applied to this as a model system. Okay, so for the next few minutes, what I want to do is show you what kinds of experiments we do to look at animal behavior. So, we're interested in the brain, and so, when things go wrong with the brain, what do we have? We have anxiety, depression, schizophrenia. How do you know if a mouse is depressed? You can't ask it. It sulks in the corner of the cage. They don't do that. In fact, mice don't ever show pain in any way because they're a prey species, and so it's in their interest to not show that they're in pain so that they won't be eaten by whatever the predator might be. And even if you do surgery on an animal, under anesthesia, you provide analgesics. Once they come out of anesthesia, they're running around the cage like nothing is wrong, whereas people after the same surgery are laying in the bed for several weeks after during a recovery period. It's really, really amazing. Okay, so I'm going to show you a couple different kinds of behavioral tests. So the first is looking at locomotor activity. So obviously when people have a stroke, they may have some loss of movement, right? And you can see that they can't walk as well or they can't speak as well. And excuse me. And sometimes it's very subtle and very nuanced. And they might have a little alteration in their gait, but you can't really tell necessarily that there was a stroke. And people have subclinical strokes all the time that aren't recognized as strokes because the effect on them is so minuscule. So this is a device called a rotorod. It's a rotating rod. In this case, this experiment is with rats. You put them on the rod, and it spins at a certain velocity, and you can ramp it up, and you can make it faster or slower. And the mice try and stay on, or the rats in this case try and stay on. So they go. This is a little bit challenging. It tests their coordination. It tests their balance. It tests their future career in the circus. But in any event, what you do is when you put them on here, you measure the amount of time it takes until they fall off, until they can't perform the task anymore. And so if you have normal mice, which are called wild-type mice, like mice or rats that you would find in the wild, versus rats that you've given a drug, you can see whether that will improve or hurt their performance in this task. Okay, so it's just looking at their ability to move, their coordination. Okay, so here's a test for depression, and we can talk about this for a minute. This is called the Porsolt Force Swim Test. You put a mouse in a big 4-liter container with water, and the mouse can't get out, and it tries to get out, and it swims around and swims around. And this is a model of learned helplessness, that after swimming for a while, it realizes it can't get out no matter what it does, and it just gives up, right? So that's what's shown here. There's a few foot flutters, but it basically says, I'm done with this. I know I can't get out. I'm not even going to try anymore. So I say that this test is a model of depression. We don't know why the mice stop. Maybe they're tired. Maybe they're tired. Maybe they're bored. Maybe they're super smart and say, this is silly. I know I can't get out. I'm not even going to try. So why do I say that this is a test for depression-like behavior? The reason is this is the #1 test that's used by the pharmaceutical industry to test new drugs that may act as antidepressants. So if you put the mouse in and you measure the amount of time it takes till it gives up, and then you give it an antidepressant, it will try and get out for a longer period of time. Okay, so how good is this assay at predicting the antidepressant efficacy of new drugs that are coming to the market? Over 90%. So when you try a new drug, if it causes the mouse to try longer to escape from this tank, then there's a greater than 90% chance that that will be an effective antidepressant in people. So I don't know what the mice are feeling. All I know is that this assay has predictive validity for antidepressant action in people. How do they know? So,
When you give the medicine to this rat or mouse, how do you know that this will how do you know that they're trying to save it?
so they're just trying to see. So they develop a new drug with the intention that it's going to be an antidepressant. So they take a group of mice, they measure how long it takes till they give up. Okay, then they take those mice and they give them the drug, and now they try for a longer period of time. Okay, now it's okay.
What's the correlation between, like you said, you don't know what the mouse is feeling. So how do we know that that's an antidepressant as opposed to the other gentleman said a stimulant?
So that's a totally right. So there could be a lot of other drugs, types of drugs, that would cause the animal to swim for a longer period of time. You could give them caffeine, you could give them amphetamine, right? So you can't put all your eggs in a basket of one test. Right, and if they were having pain and the cortisone relieved that pain, then they would swim for a longer period of time. That's possible as well. So you have this test, but there are a lot of other tests that would go along with this before you would decide, okay, this is an antidepressant. Now, for the pharmaceutical industry, it takes tens of millions, hundreds of millions of dollars to bring a drug to market, right? And many, many years of research. And most of that is preclinical research that's being done in animals. So by the time they get to clinical trials and they're trying it in people, they're absolutely sure that it's going to work the way that they think it's going to work.
Do animals ever commit suicide? Because a lot of humans do when they take antidepressants.
As far as I know, that is something that's unique to people. Well, Well, because you need a lot of animals to do this. And I don't think you can go and get, 100, 1,000 macaques out in the wild. It's just for practicality. Now, this experiment here is done with a mouse. It's better when it's done with a rat. And the reason why is because mice, if they stop swimming, they still float. Rats, if they stop swimming, they sink. So when they give up, they say, okay, I'm going to die now. I know this, but I'm just not doing it anymore. I don't know that they're thinking that. I'm anthropomorphizing. Whereas the mice, they don't necessarily have to think that. These are not my experiments. They stand in the open arms versus the closed arms. Okay, so here is a camera from above. Here's the mouse with the dot on it. It's in the closed arms right now. It doesn't actually illuminate like that. They're just doing it so you can see when it's in the open arms. And you do this experiment for 5 or 10 minutes, and then you measure the total percentage of time that they spend in the open versus the closed arms. If you give them an anti-anxiety drug, they will spend more time in the open arms. If you give them a drug that produces anxiety, they will spend more time in the closed arms. I don't know what the mice are feeling. I don't know that they're anxious. All I know is that when you treat them with drugs that have specific effects in people, you get appropriate responses using this system. Okay, so the last one is the Morris water maze. So a lot of human disorders focus on learning and memory. So obviously for people with various kinds of dementia and Alzheimer's disease, one of the things that's lost is memory. And so if you want to do Alzheimer's disease research, as far as I know, rodents don't get Alzheimer's disease. Okay, I'm live again. Okay. Rodents don't get Alzheimer's disease, but we can certainly understand about what the molecular underpinnings are associated with memory. The more we understand about how memory normally works, the more we'll be able to figure out what's going wrong in people with memory dysfunction. And so in this particular case, we're using a rat again. It's in a large pool, and you see that there's a platform underneath the surface of the water. Because it's underneath the surface, the rat can't see it. And so it just swims around. It's like a big version of the forced swim test, right? It tries to get out, and ultimately it will give up. But during the course of swimming around, it comes in contact with this platform, and it ultimately learns where the platform is. The other thing that I'd like to point out is that there are these different color or different shaped cues on either side of the pool. So when the animal ultimately finds the platform, it can figure out where the platform is in the future by figuring out where it is relative to these other spatial cues. So this is go ahead. He did find it ultimately. So this was the first trial for this animal. Right, so it takes a while. He actually swam by it a few times and bumped into it, right? But it didn't mean anything. And ultimately, at the end of this, he finds it. After you train them for a few days, the minute you put them in the water, they remember exactly where the platform is, and they swim right to it in under 5 seconds. Yeah. You can put the platform anywhere in the pool that you want. They just have to so if it were right in the middle, people would do this the same way. When you found the platform, you'd go, okay, it's equidistant from all four of these things. So they are using these other spatial clues to figure out exactly where the platform is because they can't see it because it's below the surface of the water. They can see the cues, yes. Okay, so you can train them to do this, and then you can give them drugs to see if you can improve their performance or drugs to make their performance worse. Once they're trained to find the platform, you can wait a day and see if they can still find it, wait a week, wait a year, and see do they still remember where the platform is, how good is their memory. And so this is used all the time to understand and do experiments related to memory and drugs that can affect memory, right, which you wouldn't be allowed to do in people. This is a pretty powerful assay. Yeah?
Do they typically use genetically identical animals in these tests? Because there is variation among different animals just as there is among us.
So in the case of mice, we generally use genetically identical animals to decrease the variability, right? But there are a lot of different strains of mice that are all different, and those can be used as well. And then the thing that's more interesting is we have the ability to genetically modify mice. And so if you think that there's a gene that you're interested in that's involved in learning and memory, and you modify that gene in some way, and then you can test how they perform in this task, and if they perform worse, then one possibility is that your gene is involved in memory. Okay. All right, so those are all the assays. So I'm going to spend a few minutes talking about some work that I did in my lab, but because this is a kind of a lay audience, I wanted to make sure that we were all on the same page. So in the brain, the fundamental cell type that's doing everything that we recognize that the brain normally does is called a neuron, and it's got a cell body, and then it's got a bunch of dendrites coming off the cell body, and then it has a large axon, which then has projections where it makes contacts with other cells. Okay, and it's this contact with other cells that's responsible for the cell-to-cell communication. So this is just a schematic drawing of a neuron. Here's an actual neuron. This is fluorescent because this neuron expresses a protein called green fluorescent protein, which was originally isolated from jellyfish, but you can get mice to express it, and now they have fluorescent neurons, and you can see the similarities between that and the one I've shown you here. Okay, but what we really want to do is focus on the contacts that one neuron has with the other. Okay, so here's a blow-up of that, and what I first want to point out is here's the long axon that's sending projections, and here's the contact with the next neuron right here. They don't actually touch each other. They come very close to one another, and this gap in here is called the synapse. So this is the presynaptic neuron. This is the postsynaptic neuron. And communication in the brain is electrochemical. So there's an electrical component and there's a chemical component. So the signal comes down the axon as an electrical signal. But once the electrical signal gets here to the end of the axon, it causes these synaptic vesicles that are filled with neurotransmitter to release their contents into the synapse. And these neurotransmitters diffuse across this very short space and bind to specific receptors on the postsynaptic neuron. And when these bind, they cause a signaling cascade that causes this postsynaptic neuron to do something, generally generate an electrical signal so that it can communicate with the next neuron. And so that's how you have circuits in the brain from one neuron to the next. It's electrochemical after electrochemical. Okay, so there are a number of neurotransmitters in the brain. Glutamate is the major excitatory neurotransmitter in mammals. Gamma aminobutyric acid, or GABA, is the major inhibitory neurotransmitter. Some that you've probably heard of, acetylcholine, or dopamine, or serotonin. So what I want to do for the next few minutes is talk about some work in my lab that involves serotonin. So here's a sagittal section of the mouse brain. So if I cut right down and split the brain into two hemispheres and then look at the brain on the side, this is the sagittal section. Here's the front of the mouse. Here's the back of the mouse. And so we're going to talk about serotonin. The chemical name is 5-hydroxytryptamine. Here's how a chemist would show you the structure of serotonin. And serotonin is made in a set of neurons in the brainstem called the Raphe nuclei that send projections to virtually all regions of the brain. So there's serotonin everywhere, but it's only made in a very specific part of the brain. And when it gets to its target, when it's released from neurons, it binds to receptors to cause an effect on the postsynaptic neuron. Well, serotonin is such an important neurotransmitter that in humans, there are 15 genes that encode different kinds of serotonin receptors that all signal differently. Okay, so in your genome, you have 15 different flavors of receptor that all specifically bind serotonin. So I'm going to focus on the 2C subtype of serotonin receptor. And the structure of this, the topology of this receptor is shown on this slide. And for non-scientists, you're probably saying, what is that snake? So the receptor is a protein. It's made out of amino acids, a chain of amino acids. Here's all the amino acids here. This is the plasma membrane. So this is the outside of the cell. The plasma membrane of all cells is made out of lipid. It's basically made out of fat. And this receptor goes in and out of the plasma membrane 7 times. Right? It's designed to work that way. And when serotonin binds to the receptor, this receptor changes its conformation. And when it changes its conformation, it can interact with molecules inside the cell to propagate a signal. So here's the example. Here are the intracellular molecules. They're called G proteins. It's not really important. When serotonin binds, it changes its conformation. It can interact with this G protein that then can activate a bunch of intracellular signaling pathways, which aren't really important, except to say that signaling through this receptor controls feeding, depression, anxiety, schizophrenia, seizures, and sleep. So clearly, it's an important receptor that binds serotonin. So because biology is complex, there's actually not one flavor of this receptor, but the cell can make 24 different flavors that differ by only 3 amino acids here in this region of the receptor. So you have 24 different receptor isoforms, and you look in different brain regions, and you have different combinations in different brain regions that signal differently. Because you need, in this part of the brain, you need this kind of signaling, but in this kind of the brain, through the same receptor, you need a different kind of signaling. So our question was, instead of having 24 isoforms, what if we made a mouse that only had 1 isoform? What would happen to the mouse? So we made a mouse that could only make what's called the VGV isoform, where these 3 amino acids are valine, glycine, and valine. And now they don't have 24 flavors, they only have 1 flavor. And what happens? Before I show you, I have to tell you about a human genetic disorder called Prader-Willi Syndrome. So this was a syndrome that was discovered in the 1950s. And the main characteristics are failure to thrive during the first year of life. Generally, these children don't survive unless you give them nutritional supplementation. And in this case, the supplementation is feeding through either a nasogastral tube or directly through a gastric feeding tube. If you get the kids to live for the first year, Usually then they'll survive. But sometime between the age of 2 and 6, it's like somebody flipped a switch. And now for the rest of their life, they are hungry all the time. They will eat everything in sight. And if uncontrolled, can lead to morbid obesity, where you see in this 12-year-old boy that weighs in excess of 400 pounds. There's no treatment for this. They're starving all the time. It doesn't matter how much food they consume. They are never satisfied. So whatever the machinery is, that after you've eaten food, it tells you you're no longer hungry, that does not operate in these kids. So there are some other characteristics of this disorder. Poor muscle tone early in life, short stature, cognitive impairment. They have a mean IQ of about 70, which is less than children with Down syndrome. And, but this is a rare disorder. It's only about 1 in 20,000 people. And it's caused by an anomaly on the long arm of chromosome 15. We know exactly where the deletion is on the paternal allele, the father's chromosome, that gives rise to this disorder. Okay, so why did I show you that? I'm going to argue that mice that only express one isoform, the VGV isoform of the serotonin receptor, are a mouse model of Prader-Willi syndrome. We didn't know this going in, right? We just wanted to see what would happen, and I'll show you what happened. So here is a picture of 2 mice. So remember, wild type means like found in the wild, the normal control. It doesn't mean like they're crazy. Okay, so this is at postnatal day 21. So these animals are 3 weeks old, and the normal mice weigh 15 grams. But this mutant mouse only weighs 4 grams. All right, so it's much, much smaller. And one of the characteristics of Prader-Willi syndrome is short stature. Well, mice don't have a stature because they don't stand upright, but they have a body length, right? And so they're significantly shorter than their wild-type littermates as well. Okay, so here is at postnatal day 21. These are growth curves. So this is how much they weigh in grams. And this is male and female mice in different colors. And the other thing I need to tell you is the serotonin 2C receptor is on the X chromosome. So females have two X chromosomes, so they have 2 copies, and males only have 1. So here's control wild-type males and females at the top. That's the normal growth pattern. So at postnatal day 21, they get up near 15 grams. Here at the bottom is females that have 2 copies of the mutant gene, or males that have 1 copy of the mutant gene, and they hardly grow at all. And then interestingly, right splitting the difference is females that have 1 good gene and 1 mutant gene. Okay, so you get an intermediate effect. So there's lots of ways to become small, right? And in Prader-Willi syndrome, it's because they don't eat. So we want to measure, do these mice eat less? But postnatal day 21 is when these pups are weaned from their mothers. So the only food that they get prior to that is milk from their mother. And so the question is, how do you measure how much milk a pup consumes? Right? It seems like it would be an easy experiment, but in fact it's not. So we bred the mice in a certain way to get these 3 wild-type mutant males and females that have a wild-type gene and a mutant gene, and then we injected them with water, just plain water, but in this case, the hydrogen was a heavy isotope of hydrogen called deuterium. And here, because normally hydrogen has a molecular weight of 1, and here the hydrogen has a molecular weight of 2. And here we injected the pups with water, but it had the heavy isotope of oxygen, O18. Normally oxygen is 16. So we inject them both. We wait different periods of time. We sacrifice the pups. We collect their blood. And now we basically measure how much of the heavy hydrogen that started out in the dams ends up in the pups. The only way they can get that heavy hydrogen is from the mother's milk. Okay, so when we do that experiment, you can actually calculate how much milk they intake in grams per day, and what you see is that the wild-type mice drink the most milk. The mutant males that only have one X chromosome, and it's the mutant chromosome, have the least, and those females that were in the middle that had a wild type and a mutant, they eat an amount of food that's right in the middle. So that's completely consistent with this failure to thrive. After weaning, when Prader-Willi kids start to eat uncontrollably, even though these mice are small and had this initial failure to thrive, after weaning, they eat more than their wild-type litter mates. Okay, even though they're smaller, they eat a lot more. So they go from failure to thrive to overeating, very much like the human disorder. The last experiment I'll show you for these guys then is measuring muscle strength or hypotonia. And this device is not a torture device from the Spanish Inquisition, but it is called a grip force meter. And basically you take a mouse by the base of the tail and you drag it across this grid, and the mouse's natural inclination is to grab on for dear life. And you can pull on the mouse and figure out how much force does it take to pull the mouse off the grid, okay, as a measure of their grip strength. And you can see when the mice are young, the mutants perform much less well than their control litter mates. But by the time they reach adulthood, they're fine, which is exactly what you would see in the human disorder. So I'm not saying that alterations in 2C expression, serotonin receptor expression, are the molecular basis of Prader-Willi syndrome. I can't say that with certainty. That's the hypothesis. And now we're continuing to do additional experiments to address that more directly.
So do those mutant mice become obese also?
They don't become obese. That's an excellent question. So the reason they don't, so they overeat, but they don't become obese. And the reason why is they also have an increase in their metabolic rate that offsets their increased food consumption. So that's not what happens in the human disorder. So you might say, well, maybe this isn't a good model for the human disease, but you have to realize that humans and mice regulate their metabolism in very different ways. So you can't, there aren't going to be parallels all along the syndrome. Yes?
What did you say the expected lifespan for a human with this disorder was?
If you control the comorbidity associated with the obesity, they can live a normal lifespan. So because the kids, because they grow up and the IQs are only 70, they can't take care of themselves, so they either have to live with their parents or live in group homes. And really the only treatment right now is to limit their access to food, right? So if you limit their access to food and give them a defined number of calories, then you know, they'll have a normal body mass and they'll have the normal lifespan. Go ahead.
Do they actively seek food wherever they go?
They will tear apart every cabinet and refrigerator that they possibly can to get food. I will say, let me see where we are on the time. Okay. I went to a meeting, a scientific meeting. There was a scientific session about Prader-Willi syndrome, and then there was a session with families that were learning how to deal with the disease and manage their children. Okay. While I was in the hotel, there were vending machines throughout the hotel. So they took the food out of all the vending machines. They covered them with plastic. They wrapped them with chains and padlocks. And still, every single one was broken into the next day. Because their drive to eat is so great that they will do anything they can to access food. They will eat food out of the trash. They will They will eat a lot of things that you and I would not consider palatable. Yeah. So as far as I know, it's not due to absorption of nutrients. It's the fact that early on they just refuse to eat, right? And I don't know why they don't eat. You know, maybe the muscles that allow them to suckle are defective, and I haven't figured out how to measure that yet, right? I mean, you could imagine some device that could measure the vacuum that the babies or the pups could generate, but that doesn't exist yet. So we have recent data to suggest that there are changes in serotonin receptor expression in Prader-Willi syndrome that would be consistent with the increased food intake. So we're following that up now. Because it's a rare disorder, we have a limited set of brains that we have access to, but in all the ones that we saw, there was a significant change in the expression of this receptor. Okay, so I was actually going to do a whole other thing, but because there was lots of interaction, which I encourage, I'm just going to, if you bear with me for a couple more minutes, I'll do this quickly. We're till 12:15? Then fine, I have you trapped. If I can get my computer to behave. Okay, so the last little bit, so now I'm changing my hat from Dr. Amazing or John Q. Scientist to my role as chair of the Institutional Animal Care and Use Committee and director of the Office of Animal Welfare Assurance. And so the question that I want you guys to feel comfortable, there are a lot of people that are against biomedical research with animals. I think when you think about it, there was a dark past when some things that were done to animals that I don't think anybody is too proud of, but things have dramatically changed over the past 40 years. So the question is, what about the welfare of the animals that are used in research? Is that something that we care about? And right at the outset, let me say that of course we care about the welfare of the animals, even if we didn't care about the animals themselves. We care about the welfare of the animals because when we're doing our experiments, if the animals aren't healthy, if the animals are sick, we may get a different outcome. And so just for the quality of the science alone, we want the healthiest animals that there possibly could be. Now, I'd like to think that scientists are also humans and care about other creatures and care about their welfare above and beyond the quality of the science that can be generated. Okay, so I'm going to give you a little short history of the laws in the United States that protect animals. So prior to 1966, the only law that existed to protect animals was called the 28-hour law. This was a law that was primarily related to the interstate transport of farm animals, primarily cows, pigs, sheep. And it's called the 28-hour law because when you pack all these animals in a truck, every 28 hours, the animals had to be let out to roam around for a little bit before you packed them back up again. So that was the only law that existed that looked after animal welfare. There were also the Lacey Act put into place a law which protected a number of wildlife species. So in this example, bald eagles. But there were no laws to protect animals used in research. In 1950, there was this entity called the Animal Care Panel that was developed in Chicago. And this was the first group of people that started trying to come up with guidelines for the proper care and treatment of animals. But nothing really happened from the animal care panel. And then in November of 1965, in Sports Illustrated, of all possible magazines, there was a story about Pepper the Dalmatian. So this is actually not Pepper, it's just a Dalmatian. And in this story, there was a family living in Pennsylvania, not too far from New York City, and one day their family pet Pepper disappeared. And they spent a lot of time trying to figure out, they put up signs, have you seen Pepper? Ultimately, they were able to find Pepper, had been purchased by a medical institution in New York City, used for biomedical research, and euthanized. Okay, so the public was furious. Now you're kidnapping our pets and you're using them for biomedical research. Well, the scientist at the institution bought the animal from a dog dealer, right? So they didn't know that this was a kidnapped or a dog-napped animal. So it'll probably not be a surprise to any of you. Congress was just as dysfunctional 50 years ago as it is now. So they met, they talked about this, and they introduced a bill to indicate that dog and cat dealers had to be licensed and inspected by the USDA, and of course it didn't pass. Okay, so that was in 65. In 66, there was a story in Life magazine, and I don't know if I was only 8 years old at the time, but I have a recollection of seeing this, and this is the main story in that edition of Life called Concentration Camp for Dogs. And this was a follow-up investigation at animal dealers that described neglectful and indescribably filthy conditions. So there was a huge outroar from the public, and finally Congress listened, and Congress passed the Animal Welfare Act in 1966. This required the licensing of animal dealers, minimum standards of care for dogs and cats, and they were subject to unannounced inspections either by the U.S. Department of Agriculture or the Animal and Plant Health Inspection Service. Since 1966, this Animal Welfare Act has been amended 6 times. Okay, so as a consequence of the Animal Welfare Act, an institution such as Vanderbilt must obtain a license from the USDA. We have to meet minimum standards for animal care. We have to obtain animals only from registered dealers, not some fly-by-night, here's a beagle, I can get you for a cheap price. They have to develop an institutional animal care and use committee, or IACUC, to oversee all the animal research that's being done. They have to minimize the pain and distress that are provided to the animals during experimental research. They have to consider alternatives to these painful procedures and use them when possible. And then they have to provide animal enrichment. So you can imagine just sitting in a cage day after day can be pretty boring. You see the animals pacing back and forth at the zoo, largely just because they're bored. So the animals have to be provided enrichment to make their environment interesting. In the case of zebrafish, you put some extra seaweed in the tank, and they seem to really like that. In the case of mice, you give them little rolled-up pieces of paper, and they will spend the whole day just unrolling it into shreds all day long. But for something like the non-human primates at Vanderbilt, they watch movies, they listen to music, they play with toys, they have human interactions with people multiple times a day. So I think that's pretty good. It depends on the movies and the music. But one of the things I want to point out is that the Animal Welfare Act didn't include all species. So it's dogs, cats, non-human primates, guinea pigs, rabbits, right? But it didn't include birds, mice, rats, horses, or any other farm animals that would be used for food. So the way that hogs are treated, hogs are slaughtered, that chicken are treated, that are provided for food that you would pick up at Kroger, the quality of that animal care is not regulated by the Animal Welfare Act. And I think if you watch any TV at all, you see that those are pretty bad conditions in some instances. So, shortly after that then came the Public Health Service Policy. So this was a separate set of guidelines that is put into place for all institutions that are supported by any PHS agency, either the National Institutes of Health, which funds most biomedical research, has to conform to these guidelines, the Food and Drug Administration, so now that covers all of the animals that are used for food production, and the Centers for Disease Control and Prevention. Anybody who's doing anything related to these has to also follow this. So at NIH, there's the Office of Laboratory Animal Welfare, OLAW, who puts all this into place, and they created this thing known as the Guide. It is basically the handbook for all the federal regulations for how these animals are to be treated. It's the Guide for the Care and Use of Laboratory Animals. And this covers any live vertebrate animal used or intended for use in research training, experimentation, or biological testing. So the public health policy, or the Public Health Service Policy, requires an IACUC, procedures for self-monitoring, that the IACUC serves as the local policeman to make sure that all of the investigators in the institution is compliant with all of the federal regulations, an occupational health and safety program so that people don't endanger themselves by working with laboratory animals, a training program so that people, when they're working with the animals, they know what they're doing, environment, housing, and management program. This is the enrichment program that I told you about, and that they appropriately maintain the facilities for housing and support. So here's the IACUC. Its overall goal to ensure the humane care and use of animals used in research. And according to the Guide, there only have to be 5 members on the IACUC. The chair, which is me in this case, the attending veterinarian, a scientist member, a scientist who's not in a biological field, and most interesting, the non-affiliated member. So the non-affiliated member is called the community member. This is just someone out in the public who has agreed to serve on the IACUC. Currently, we have 2 community members. One of them is a stay-at-home mother who had some previous biological training long ago when she was in college, and the other community member is a pastry chef. So, who has really no knowledge of biological research, but she lives on a farm and has a lot of animals, particularly horses on the farm. So she has very strong opinions about animal research. So we don't need anybody right now, but in the future, if someone would like to serve on the IACUC, it's really a commitment of 4 hours a month and a little bit more. We would be happy, if you're interested, to talk to you, tell you what's required, because it's really the input from the non-affiliated members that's important. Because I see the research that's getting done all the time, I sort of get jaded, right? I see, yes, you're doing this to this animal or another. And it's the community members that really have, why are you doing this? What good does this experiment, what is the advantage of this experiment? How will this advance human health? Why are they doing it with 10 animals? Why couldn't they just do it with 5? So the community members are really critical to this whole process. Okay, the last thing then that we try and promote is what's called the 3 R's of using animals in research. The first R is replacement. So if you're going to do a scientific experiment and you think you need animals, is there some other way? Is there some other system that you can use to not use animals? Can you use a computer program? Can you use, some kind of robotic dummy, right? Which a lot of people are doing for a lot of training. At Vanderbilt, for example, they do training with ferrets. And the reason they use ferrets is they intubate the ferrets because the size of the ferret trachea is the same as a newborn baby. And so in the pediatric wards, when they have to intubate the babies, they want to make sure that these physicians have gotten proper training. And so I would argue, if you had a newborn baby in intensive care and it needed to be intubated, would you like it to be done by a physician who's actually had hands-on experience using a ferret or somebody who did it on a computer? And theoretically, they know how to do it, but they've actually never done the procedure themselves. The second R is reduction, to use as few animals as possible. Each investigator has to write an animal protocol that's approved by the IACUC that specifically says how many animals you're going to test. If you say you need 10, you have to justify why 10, why not 8, why not 3. And there are statistical tests that you can employ to say, I've used these statistical analyses, and 10 is the least number that I can possibly use. And the final one is refinement. So we're constantly trying to improve the procedures to induce less pain and distress in the animals. 20 years ago, there would be a certain kind of procedure that's done that would cause a certain amount of distress. And now we've improved our techniques, and so the distress, the pain is less. So we encourage the investigators to use the most refined techniques to cause the least pain to the animals possible. Yes? It's possible. It's an always changing target. So each year, as new technologies become developed, right, we're constantly looking at the 3 Rs, right? And so now that there are 3D printers, it's totally possible that you could make, something that would be the appropriate size to intubate, and then it would be up to the investigator to explain why they couldn't use that instead. Okay, so the last thing that I want to say then is kind of the take-home message is good science and good animal care go hand in hand. The better the quality of the animal care, the less pain and distress we inflict on the animals, the better the quality of the scientific data. So I'd like to thank my colleagues, I'd like to thank the people in my lab for the little bit of data that I provided for you, and I'd like to thank my colleagues at the Vanderbilt Brain Institute, the Center for Integrative and Cognitive Neuroscience, the Kennedy Center for Research on Human Development, the Silvio Conti Center for Neuroscience Research, the Vanderbilt Vision Center, and the Vanderbilt University Institute for Imaging Sciences. And with that, I'd be happy to take any other questions you might have. Yes? Okay. So the question is there something about jellyfish, some protein from jellyfish, that can improve memory? I don't know the answer to that question. But now I'm going to go back and search for it and see if I can find that. All right, well, thank you very much.